Clinical microbiology, the study of microorganisms in a clinical setting, focusing on the diagnosis and treatment of the infections. For example, identifying the Streptococcus pneumonia in the patient samples to diagnose pneumonia. Actually, we have to study the steps that actually happens in the clinical settings. So there are actually three main levels that we can go through to identify the microorganisms. Now, the first one is the microscopy. Of course, before doing everything, we have to go to the microscopy method, right? We just have to take the sample of the microorganism and then we have to go to the staining, like coloring it, since we can't see invisible stuff, right? Or maybe it's fungi, so we don't have to color it. So anyway, we color it, or in scientific term we stain it and then put it on a slide and under the microscope, and we study its structure, physical structure. Actually, before microscopy we have to grow them, culture them, and then we have to study its physical appearance. Like in case of bacteria, if it grows on the plate, agar plate, the one that we have grown on, it creates colonies. The space where the bacterial cells are congested is actually the colonies, and it becomes colonies only when the same type of bacteria are close. So we can study the bacteria from colonies. Some bacteria creates larger colonies, some creates colonies size of a dot, or some bacteria has colonies that are concave, some has convex, and some are puffy, and some are flat, and some are colorful, like yellow, whitish, or greenish, brownish. Like we can actually categorize them based on their colors. Like we can shorten the spectrum of our diagnosis. After that, we stain them. And then under the microscope, we study more of their characteristics on cellular level, like their cell wall, or they have flagellates, or or what kind of organelles they have, or what kind of nucleus they have, or things like that. After these things are done, and we still are not sure what kind of microorganism this is. I mean, we have shortened the spectrum, like its shape is rod shape, so it must be bacilli. But what kind of bacilli actually? So after that, we go after the chemical methods, and in that we do the catalase test, oxidase test, triple line test, and tests of, like, there are multiple. There is actually a list of that, and we will cover that in another video sometime.